Hello, everyone. So I can see lots of students are joining us very promptly. So thank you so much, everyone, for joining. So my name's Freddie. I'm here from Team Unifrog, and it's really great to see so many of you joining us this evening. I will wait just a few seconds for more of you to filter through. I can see there's about 80 of you plus that have joined. So we're really ecstatic to have all of you here today. And as you can see, we're going to be having a great sort of lecture style subject taster session from Andrew Young, all about understanding schizophrenia. And we're very lucky as well to be joined by Sumayyah, who is a student at Leicester, currently on a placement year. And we're eager to hear how your journey's been and maybe fire some questions at you, Sumayyah, towards the end as well. Hopefully that's okay. Just on that note, um, we do have the Q&A button, which I'm sure a lot of you will be familiar with from previous Unifrog webinars. So throughout today's session, if you've got any burning questions at all, please do click on that button, post your questions. And Andrew has assured me that the last sort of 10, 15 minutes, we're going to have lots of time for Q&A and open discussion. So we're really looking forward to having your interaction and input as well. Without further ado then, because we don't have too long, I'm going to hand over to Andrew to take it from here.
Hello, thank you, Freddie, and welcome everyone to Leicester, albeit electronically rather than in person. As Freddie said, I'm going to be talking about understanding schizophrenia from behavior to brain function. I do want to qualify something that Freddie said, though. I'm not going to be telling you all about schizophrenia, but I'll be giving you a taster of it. So, as I say, I'm Dr. Andrew Young from the School of Psychology and Vision Sciences. So, why have I chosen to talk about this? Well, schizophrenia is a psychiatric disease which is of general interest to many people. Many people know a bit about it, but not many people know much about it. Not many people know about it in detail. But importantly, it illustrates the synergy between biological and behavioral neuroscience and psychopathology and many other areas of psychology, cognitive neuroscience, cognitive psychology, individual differences, and it gives a flavor of some of the methods being used. So it's quite a nice topic to look at in that sense. It's also a topic that I've spent my career researching, so it's coming straight from my research interests. Okay, so we were able to use this to understand a poorly understood psychiatric disease and inform future clinical practice. Now, one of the purposes of this talk is to give you a sample of teaching across the 3 years of our undergraduate courses and how that links with current research. So most of the material is taken from lectures currently given to our undergraduate students, and this is flagged up by the green, blue, and red circles on each slide. So the green, blue, and red circles, and also the research one, which goes beyond the teaching side of things and looks at some of the current research. And those will appear up in the top corner, just to give you some sort of guide as to what sort of things appear on the course. Now, psychology as a discipline, as you're probably aware, has many different facets, and they're just summarized here. And one of the things I just mentioned is that this talk really looks at the synergy between a number of those, and I've just highlighted there the areas that we'll be looking at: cognitive, individual differences, psychopathology, biological and cognitive, and applied, of course, because we're looking at a clinical condition, and also looking at the methods of investigation. So first of all, looking at the psychopathology of schizophrenia. So thinking about how we understand it, I'm going to be talking about what we do know about schizophrenia, where I'm also going to just mention what we don't know about schizophrenia, and look at why schizophrenia is so difficult to study. And I'm then going to take that on to look at modeling schizophrenia in animal models in order to gain an understanding of dysfunction and of treatment. So, looking at the psychopathology of schizophrenia then, what is it? It's a psychotic illness that affects round about 0.5% of the population. The peak onset is in late adolescence and early adulthood. So I'm still having trouble with advancing the slides. So we do know at least something about the biological basis and structural and chemical abnormalities, but really it's very uncertain. And for example, we know that in many schizophrenia patients, they have enlarged ventricles. These are fluid-filled spaces in the brain. And you can see here in a normal, this is an MRI scan of a normal brain, and you can see the dark patch there is the ventricle. And you can see in the brain from a schizophrenia patient, you have a much enlarged ventricle. There's also reduced brain tissue volume, and you can see this quite clearly in an area called the hippocampus, just here, where you can see a very much reduced tissue volume. So this occurs in schizophrenia patients, but not in all schizophrenia patients. So it's certainly something that's common in schizophrenia, but it's not a sort of an indication of schizophrenia. People with enlarged ventricles don't necessarily exhibit schizophrenia, and people with schizophrenia don't necessarily have enlarged ventricles. So it's giving us some clues about the biology, but it's not absolute. Now, we also know that a number of brain chemicals change in schizophrenia, and there's 3 of them here. There's dopamine, serotonin, and glutamate. And I'm going to be focusing primarily on dopamine, but I will be bringing in serotonin and glutamate later. And of course, these are the ones that are really important in terms of treatment, because all our treatments that we have now are aimed at brain chemicals. All the pharmacological treatments are aimed at brain chemicals, mainly dopamine, but some serotonin as well. But we'll come to that later on. The symptoms of schizophrenia are odd as well, because they differ between patients. And it's a common feature that different patients with a diagnosis of schizophrenia may actually show very, very different clusters of symptoms. And they also fluctuate within patients. So from month to month, one patient may exhibit very different sorts of symptoms. Now, there are 3 main types of symptoms, and the positive symptoms are essentially an inability to differentiate a person's own thoughts and ideas and perceptions and imaginings from reality. And this results in things like hallucinations, mainly auditory hallucinations, delusions, and what we call disordered thought, where thought processes are not in a logical sequence. Negative symptoms are essentially a damping down of normal symptoms, a lack of interest, emotional flatness, so not responding to emotional stimuli, and social withdrawal, and becoming very much withdrawn into oneself. And then cognitive symptoms, dysfunctions in things like memory, attention, and cognitive flexibility, the ability to change your cognitive processes in response to environmental changes. Now, these are mainly diagnosed by asking a patient what they are experiencing, and this immediately gives us a problem in trying to look at this scientifically, because that's a subjective measure, and we need an objective measure. We need to measure objectively what's happening in order to study this scientifically. And therefore, it's very difficult to study the mechanisms of the disease. What about treatment? I mentioned it briefly. Well, it's really not very good, and it's very slow in onset. So this little diagram shows within a month of starting treatment, more than 3/4 of the patients will show little or no recovery. And then there's very small amounts of recovery, near full recovery or substantial recovery, and some recovery, but not much. Once that goes up to a year of treatment, you can see that it's very much better, but it's still not all that good. You've got round about 1/4 of the people showing near full recovery, another 1/4 showing substantial recovery, but still requiring specialist care, a 1/4 showing some recovery, but little, but still hospitalized, and nearly a 1/4 showing very little recovery at all. That's after a year. So actually the treatments that we have are a lot better than we had before they were brought in, but actually they're not very good. And Peter McKenna, who wrote an influential book in 1997, said its symptoms are numerous, its presentations are diverse, its cause is unknown, and its response to treatment is unsatisfactory. Now, that was written 25 years ago, and actually the case is not much different now. So we know a little bit more about it, but we still don't really understand what's going on. So how can we understand better what's going wrong in schizophrenia? How can we treat schizophrenia more effectively? Well, this is where psychology comes into it. And we can start to model behavioral dysfunction in animals to study the underlying mechanisms. We can look at things that are going wrong in schizophrenia and look at them in animal models. We can't model schizophrenia as such, partly because schizophrenia is diagnosed through talking to the patient. And you can talk to rats as much as you like, but you don't often get an answer. But importantly, when you're doing animal experiments, you must be able to link those animal experiments back to normal and abnormal human function. And that's where I'm going to go just now. Now, if we look right back in history, Kraepelin was the first person to give a formal description of the symptoms of schizophrenia. And what he said was schizophrenic patients are unable to exclude irrelevant sensory information from their awareness. And so that tells us something about what sort of behavioral abnormalities are there. And it may be indicative of deficits in selective attention. That is, our ability to focus on what's important. You can filter out what's going on in the background and focus on the important stuff. And this is where cognitive psychology comes in, because we know of behavioral tasks which measure selective attention in people and in animals. And the one I'm going to talk to you about is latent inhibition. Now, you've probably all come across conditioned learning, Pavlovian learning, and this is the case where a stimulus, such as a bell, and where it's paired with food, and subsequently, when the bell is rung, you get salivation, as in Pavlov's dogs, or you get an approach behavior to the food, and that shows good learning. Now, we're calling this NPE, non-pre-exposed, because what's happening in latent inhibition is if we pre-expose the stimulus before this pairing, then it impairs its ability to enter the conditioned association. So showing that diagrammatically, we're giving lots and lots of rings of the bell with nothing happening. Then we come to do the pairing exactly the same as above, but because they've learned that the stimulus is irrelevant, they therefore don't learn this association between the bell and the food. And that is latent inhibition, the difference between the learning in these two conditions. Now, I've shown these in red and blue because that's how we're going to depict it in the diagrams coming up. So the red is the pre-exposed, where they've had this repeated pre-exposure, and they're showing very bad learning. And the blue is just normal learning. So in this graph, we've got the poor learning is down at the bottom of the graph and the good learning is at the top of the graph. So here in blue is the non-pre-exposed showing good learning, and in red is the pre-exposed showing poor learning. Now, that's all very well. That's a nice little psychological task, which you can demonstrate quite simply in many different ways. Why is that of interest to us in schizophrenia? And this work comes from work from my group when I was in London, and also from a group in Tel Aviv. And that we've shown that in schizophrenia patients, this latent inhibition is disrupted. And it's disrupted in a very specific way. It's not the basic learning that's disrupted. It's the ability, it's the ability of the pre-exposure to affect that learning. So in other words, they're not using the learned information from the pre-exposure in the current processing. So that's the latent inhibition is disrupted in schizophrenia patients. And we also know that drugs which are used to treat schizophrenia. And the one the example I've given here is haloperidol, which is an antipsychotic drug that reverses this effect of schizophrenia, and it stabilizes the symptoms of schizophrenia, but it also reverses the effect on latent inhibition. So a really important thing to see. Now, a slip of the tongue there, I also mentioned amphetamine, because very, very similar effects happen with normal people given the drug amphetamine, which I'm sure you've all heard of amphetamine. And so amphetamine causes this reduction of latent inhibition, and that is reversed by the same drug, haloperidol. So summarizing that, schizophrenia patients learn well even after pre-exposure. The change in latent inhibition is reversed by haloperidol, and similar changes are seen with the drug amphetamine. Now, what do these drugs do? Well, haloperidol is an antipsychotic and it affects dopamine signaling, and amphetamine also affects dopamine signaling. And it's these two observations that have led to the dopamine theory of schizophrenia. Now, I'm just going to go off on a slight tangent here to explain that in a little more detail. And this is where we get into sort of the real biological psychology and what's happening in the brain. So here's a picture of a synapse. It's where one neuron connects to another. And neurotransmitters, these chemicals, are released from one neuron into a space between the neurons, and then they interact with receptors on the next neuron in the chain and transmit the signal. And what these drugs are doing, amphetamine increases the amount of dopamine here, so it's increasing dopamine signaling. So increasing dopamine signaling is producing something that looks like schizophrenia. Haloperidol is an antagonist at these receptors, so it sits in the receptors and prevents the transmitter from binding. So it's reducing the dopamine function. So by reducing the dopamine function, we improve schizophrenia symptoms, and we also restore latent inhibition. So that was very quick there. Now, we can also show this in experimental animals, such as rats. And the same drugs disrupt latent inhibition in rats, so things like amphetamine. And as in humans, they're restored by the drugs which treat schizophrenia. So we can use latent inhibition studies in rats to study the mechanisms underlying schizophrenia. Now, I haven't got time to go into this in any detail, but I want to show the principle of what we're trying to do with this. So here's the graphs that you've already seen. So the normal latent inhibition there and the disruption by amphetamine. And we can ask the question, what other experimental manipulations disrupt latent inhibition? So we know that amphetamine does, and that gives us a clue that maybe dopamine is involved. But what other manipulations might disrupt it? And can they inform us about brain dysfunctions in schizophrenia? Well, there's a number of things, another of examples I can give you. First of all, we can reduce glutamate function. I remember I mentioned glutamate as another of the neurotransmitters, and that emphasizes the importance of glutamate systems in schizophrenia. And this actually comes from our own labs, published in 2020. Asma Al-Ali did that, showing this that reducing glutamate also disrupted latent inhibition in this way. Other things, hippocampal damage. Hippocampus is an area of the brain, and we'll come to it again in a minute, but it's a very specific area of the brain which seems to be involved with schizophrenia. You saw it in an earlier slide where I showed that there's a reduction in tissue volume in the hippocampus. So there's implications that the hippocampus is involved. And as I say, we'll come back to it again. So again, damage to the hippocampus is also disrupting latent inhibition. This is an interesting one from some of our colleagues just up the road in Nottingham, where separation of the rat pups from their mother for an hour a day during the pre-weaning stage caused a similar sort of disruption. And this is modeling the effect of upbringing. And we know that that upbringing has an effect on schizophrenia, and this models it. And so it's in a way you're bringing in developmental psychology here as well now, but you're able to model the effect of the upbringing in causing schizophrenia-like changes. Now, remember, I said schizophrenia-like changes. We're not causing schizophrenia here. We're causing changes in behavior which relate to schizophrenia. So that's how these sorts of studies can inform us about brain dysfunctions in schizophrenia. What else can they do? Well, we know that haloperidol, which is an antipsychotic drug, we know that it reverses this effect of amphetamine. And we can ask the question, what other drugs might restore latent inhibition after amphetamine treatment? And can they help us discover potential new antipsychotic drugs? And I can illustrate that because drugs which act at both dopamine and serotonin systems have led us to some of the newer antipsychotic drugs. Haloperidol is not used very much now. It is still used in some patients, but the newer antipsychotic drugs act on both dopamine and serotonin systems. And I just want to take a little diversion away for a minute and show you some experimental data. And this comes from Felden's group in Tel Aviv. And you can see what haloperidol does. But if you give a low dose of haloperidol, it doesn't have an effect. It doesn't abolish the effect of amphetamine. So it needs a high dose in order to have the therapeutic-like effect. However, if you give, and also if you give a serotonin antagonist, it doesn't have any effect either, or a very minimal effect. However, if you give those two together, that low dose of haloperidol and that same dose of the serotonin antagonist, you do see the restoration of latent inhibition. And that's very interesting because modern antipsychotic drugs work at both dopamine and serotonin systems. And this is highlighting the importance of the dual mechanism. Okay, so going back to this slide again, we also know that drugs which moderate glutamate function restore the latent inhibition. And there are some novel drugs along these lines undergoing clinical trials at the moment. Now, this is important because actually we haven't had any major advances in the type of antipsychotic drugs for the last 25 years. The introduction of these dopamine serotonin drugs was around about 25 years ago. And yes, we've got different types of those drugs, but we haven't had any new pharmacological approaches. So the fact that some of these are going into clinical trials now is really encouraging. And then there's some drugs that we've been looking at affecting only some dopamine receptors. We can go into all sorts of complexity about the types of dopamine receptors, but there are essentially 5 types of dopamine receptor. Drugs like haloperidol act on the number 2, the dopamine D2 receptors. We've been looking at dopamine D4 receptors, and Pooja Sood and my current PhD student, Kalapo Teneviaji, have been looking at that. And we're getting really encouraging data from that. So there's a number of ways that we can begin to look at new types of antipsychotic drugs through these sorts of models. And as I've said, some of them are now going to clinical trial. But I mentioned earlier on, we have to be able to tie this back to the human state. And so I now want to go back to some other studies that we did quite a while ago when I was still in London, looking at normal individuals, normal people undergoing latent inhibition and doing functional MRI scanning while they were doing the latent inhibition task. And here are some sections of the brain, and the areas that are shown in yellow are areas of the brain which become more active during the non-pre-exposed than during the pre-exposed. So in other words, during pre-exposure, they become less active. Now, I don't expect you to know where these areas are, but if you're really interested, it's published in our paper with myself and Veena Kumari. But these are areas of their temporal lobe areas, including the hippocampus. And these bits that are circled are the hippocampus. So we've come across the hippocampus before, and now we have direct evidence that the hippocampus is involved in processing with latent inhibition. And we also know that these areas are suggested to be abnormal in schizophrenia, and we know that latent inhibition is abnormal in schizophrenia. So we're beginning to tie these things together. And I said, damage to these areas disrupts latent inhibition in animals. Now, the next experiment from this should have been doing this in schizophrenia patients. But if any of you have ever had an MRI scan, you'll know it's a very enclosed area. And quite simply, we cannot put someone who is suffering positive symptoms of schizophrenia into the scanner for it takes around about 50 minutes to run this. And we simply can't do it. It's not ethical, it's not practical. So we never actually managed to get that next stage of the task done. But it's nevertheless giving really nice indications. Now, I mentioned individual differences as well. And we've done a lot of research using a feature. It's an individual difference, a personality scale called schizotypy. It's a personality measure in the population as a whole. And it may link to aspects of schizophrenia. It is not schizophrenia. High schizotypy scores may indicate a vulnerability for future schizophrenia, but it's not absolute. It's not, if you have a high schizotypy score, it doesn't mean you are going to get schizophrenia. We don't fully understand the link between, which is partly why we're doing so much research on it. And we know that latent inhibition is reduced in high schizotypy individuals, and here are the data for it. And this is redrawn from a paper by Kiri Granger and myself from 2012. And so you can see that people with low schizotypy scores show full latent inhibition, but with high schizotypy scores, they show disrupted latent inhibition. So this is giving us an indication that schizotypy, this personality measure, which links to aspects of schizophrenia, is also linking to latent inhibition, giving us more clues as to what sort of brain dysfunctions are going on. Now, the other reason I wanted to show this is that Kiri Granger was actually one of our undergraduate students, and these are the results from her undergraduate dissertation research. So it's emphasizing that as an undergraduate in our department, you do research that is real research and has impact. This was published in Behavioral Brain Research with Kiri as the first author in 2012. So it's emphasizing that if you're doing a research project, you're not just playing around with made-up data. You are doing real fundamental research. Okay, I just want to draw this to a conclusion now. So schizophrenia is a condition affecting round about 0.5% of the population. Sufferers experience a dissociation from reality. It has severe effects on social functioning and relationships, often leading to long-term hospitalization. Despite this, we have a very poor understanding of its cause, and many patients don't respond well to the treatment. Remember that quote from Peter McKenna. We can use animal models to study the psychological functions which are abnormal in schizophrenia, and looking at the neurochemistry and neurophysiology of these functions, understand what's going on in the brain and what abnormalities may be underlying the behavioral changes we see in schizophrenia. And once we can use these experimental schizophrenia-like states to test this, and this will then give us a better understanding of the mechanisms underlying these behaviors in people and underlying the abnormalities that we see in schizophrenia. And these results are informing clinical practice. As I showed you, some of these studies that we've done over the last 20 years are now going into clinical trials. So these are the sorts of studies that are going to produce the antipsychotic drugs of the future. So it's not only improving our understanding of what's causing the disease, which is we still don't really understand it, but also improving treatments. So if this presentation has sparked your interest in psychology, I hope you'll think about the University of Leicester as a place to study. So just as a final couple of slides, I just want to give you a brief overview of psychology at Leicester. We have 3 degree streams on offer: a BSc in psychology, a BSc in psychology with cognitive neuroscience, and a BSc in applied psychology. The 3 degree streams they share 2/3 of the teaching material. It's only 1/3 that's slightly different, and it depends on which sort of specialization you want to do. If you do want to come on one of the courses, you don't need to decide between them before you come. You can change once you're here. All our degrees are accredited for graduate basis of chartership by the British Psychological Society, which is very important if you want to study. If you want to be a professional psychologist, you need to have a BPS accredited degree. All our degrees are BPS accredited. So whether you want to come to Leicester or not, look out for that. Why choose Leicester? Well, we've got a broad and balanced skills-focused curriculum. So we look at the skills as well as the topic. Diverse methods of teaching and assessment. We've got this synergy between teaching and research, as I hope I showed you as we went through the slides. The current research is informing the teaching, and people get the opportunity to do research. High quality teaching, that final year research project, doing real research. Diverse culture and backgrounds. Leicester is a very culturally diverse city. Highly effective support systems. Essentially, it's a degree that prepares you for a variety of careers. And I think I'll leave you with a quote from one of our graduates. I met some brilliant and inspirational individuals at Leicester, had an amazing time socially, and developed skills which have led me down a path of a successful and enriching career. She's a principal statistician at the Home Office. If you do want to come, these are our current things that were the requirements. You can contact our recruitment team on this email or via the university's recruitment team. These entry requirements do change from time to time, but this is what they currently are. So with that, I'll thank you for your attention. I'll be pleased to answer questions on my talk or about studying in Leicester. Thank you very much. So before we go on to look at some questions, I just want to introduce Sumayyah, who, as Freddie said, is one of our students. She's currently doing a year in industry, so between her 2nd and 3rd year. So Sumayyah, would you just like to say a few words, please?
Of course, yes. Hi, everyone. My name is Sumayyah. I'm currently on my placement year. I am doing psychology with cognitive neuroscience. Finished my 1st and 2nd year, working with the recruitment team at the university as a UK student recruitment assistant. And yeah, so what I do currently is like go on careers fairs and HE fairs and speak about student life at University of Leicester. And yeah, that's me. So if you have any questions about the university in general or student life at Leicester, do let me know and I'm ready to answer it.
Thank you. So I'm sure that you're keeping an eye on the Q&A coming in. I've just been looking at these and so I'll go through and I'll answer as many of these as I can. We've got about 10 minutes. How would you suggest supporting a family member who has recently been diagnosed with schizophrenia? I would like to point out that I am not a clinician. I am a fundamental neuroscientist or biological psychologist. So really, I can't give any sort of clinical diagnoses. By far the best advice I can give is to follow the advice of clinicians. But as a fundamental neuroscientist, and from my knowledge of schizophrenia and the sorts of things that someone who is experiencing schizophrenia, the sorts of things they're experiencing, I would just say try and be understanding of what that person is experiencing. And in some ways, I can't go into this in any detail, but in many cases, what someone suffering from schizophrenia is experiencing is quite a frightening world for them. And so that sort of understanding, I think that would be my main message. But as I say, I'm not clinically trained, so do follow the advice of clinicians. Where do temporary hallucinations in the peripheral of one's vision? Yeah, this is one thing that people often don't realize, that everyone experiences hallucinations from time to time. And they're nothing to do with schizophrenia. And very often when you're just at that sort of stage of almost falling asleep, you experience hallucinations. And they can be visual, they can be auditory, they can even be somatosensory. And hallucinations are actually, they're not abnormal. Most people suffer them or experience them. But I think the thing that separates them from schizophrenia hallucinations is that a non-schizophrenia person will experience those and realize that's what they are. Whereas someone who is experiencing a hallucination as part of schizophrenia doesn't have that insight that it's not real, that it's a hallucination. They actually believe that is the reality. So yeah, it's an interesting one in terms of how we process the act of a hallucination. And I think that really answers the next question as well with maladaptive daydreaming, and possibly also are delusions always linked to hallucinations, or can they exist independently? Yeah, they normally are. Someone who is having hallucinations is normally also experiencing delusions if they're suffering from schizophrenia, but it's not necessarily the case. A very common sort of delusion is a delusion of persecution, where people believe their flatmates are working for the FBI or whatever. And that very often then is associated with auditory hallucinations, where they believe the people are talking about them. They don't have to be, but they very often are. How would long-lasting injections work for schizophrenia? Right, this is the so-called depot injections. And these are where, for the most part, people who are suffering from schizophrenia need to stay on medication long-term in order to stay symptom-free. And many people don't like that, because some of these drugs, as well as not being particularly effective antipsychotics, they also have quite unpleasant side effects. So people come off the drugs, and we call that non-compliance. And very often that then leads back into another psychotic episode. So patients who are non-compliant are very often given what we call a depot injection, which is it's a preparation of many of these antipsychotic drugs, which is injected normally into the muscles, and it releases slowly over a period of a month. So rather than having to take medication daily or even more than once a day, they have an injection once a month, and it aids compliance. For the most part, these are the older antipsychotic drugs which are not such good antipsychotic drugs, simply because we don't yet have the depot preparations for the newer drugs, but they are now coming online. So we are getting those. So the newer drugs are now being used in this way. How would you summarize schizophrenia to a child who is below GCSE and ALM? Sorry, Freddie.
So sorry for butting in, but I just wondered, Andrew, just while we've got Sumayyah here, whether it might be a great opportunity just before we go, if Sumayyah is still here,
yeah,
whether we could just ask her about her time at Leicester, and and maybe just your favorite aspects of your course, Sumayyah. And at the same time, we've got so many questions coming through as well, and I know that we're just not going to be able to answer everything. But
sure.
Andrew, I wondered if, as well as this, we could also perhaps share your email address. Would that be okay?
Yeah, yes, yeah.
Yeah. Fire that across to me in the meantime, Andrew. And Sumayyah, how are you getting on?
Okay, so I would say I found one of my favorite things at University of Leicester is assessments and how that has how we get assessed basically on our modules and so on. Usually when we think of university, we kind of think of it as like a stressful time and so on. But I found that the exams over here have been quite well, basically. We get enough coursework in a sense where we have enough time to actually submit it and get them done. I'm really fascinated by the things that we learn. So I really enjoy the workshops that we get to do, the brain scans that we do in brain and cognition. There's also a lot of facilities at the university that we get to experience. Do enjoy that as well. I'd say that the modules at University of Leicester are quite unique as well. One of my favorite ones is brain and behavior module that I really enjoy. I like the equal amount of theory stuff that we do, of like essays and writing and so on, but also a lot of hands-on experience that we get to do as well. So yeah, I would say those are a couple of things I enjoy at University of Leicester.
That's really cool. And Andrew touched on the sheer amount of different sort of career opportunities he might be able to go on to, with a degree in psychology or, allied subject areas. So I wondered for you, Sumayyah, like what your plans are, if you have any at the moment, following.
Yes. Well, in the long run, I do want to work as a cognitive behavioral therapist. That's what I want to go into, but I always want to like try out different things, to be honest, things that are related to psychology. So there's, of course, the CBT and also counseling in general, getting to work with patients and so on, but I also want to do research, learn about mental health in more in depth. So yeah, I think also want to carry on doing masters and just further studies in general. But yeah, CBT probably.
That's really cool. And I guess the exciting thing is, you don't have to box yourself into one specific thing, like just following university. You've got so many options, I guess, as your interests develop and your skills develop in the years ahead, which is really cool.
Yes, I am working right now with the recruitment team, so that's amazing as well. So I can actually go into that if I do want to. Psychology does open doors to like a lot of different career opportunities.
Fantastic. We do have so many questions coming through. So I wondered, Andrew, are there any jumping out to you at the moment? Perhaps you could answer one final one, and then I'm gonna share for everyone here right now the details, the email address that you've got here, so that any follow-up questions can be asked. And Sumayyah, I wondered if students wanted to ask you anything about your student experience and your studies following this session, would they be able to do that?
Of course, yeah, I can share my email in the chat as well.
Great, pop that over to me, and I'll share that with everyone. But Andrew, what do you think? Are there any jumping out to you here in the Q&A box?
I've just been answering some. I've just been typing some answers while Sumayyah's been talking.
Amazing.
But I think, is there a leading idea for the cause of schizophrenia currently? I think that's a wonderful question. If you've got another 3 hours, I can answer it. Yes, we are getting there, and we know a lot about what is going wrong, but how that all connects up. And what we think at the moment is there's two main areas of the brain that seem to be critically involved: the temporal cortex, which includes the hippocampus, which I mentioned, and the frontal cortex, which, as the name suggests, is at the front. And for a long time, it was thought that there was some abnormality in those areas. But actually, there's a theory came out, it's quite a long time ago now, in about 2015, called the disconnectionist theory. And this was not so much that it was a normal connection between two areas that were abnormal, but rather that it was an abnormal connection between two areas that are normal. So the temporal and frontal lobes are functioning in themselves perfectly normally, but the communication between them is abnormal. And this fits in with what we also know, is that it is something that it's a neurodevelopmental disease. It's something to do with the development of the brain, and it in quite early life as well. And that all fits in with that. So yes, we're getting there, but we still don't really know the absolute fundamental cause. And without that, it's going to be very difficult to treat the cause. We are only treating symptoms. So I think that's quite a good place to end.
Fantastic. I've learned a lot over this 45 minutes. So I certainly sure that I would be joined by the sort of 100 or so students that have come along today, who I think will have learned a lot from this as well. So a massive, massive thank you to Dr. Andrew Young for delivering this, and of course as well to Sumayyah for your answers there as well. And I have popped Sumayyah's email address if you did want to follow up with any more specific questions, and Andrew's is there as well. And then we also have the psychology program areas, course areas on Leicester's website. You've got a link to that as well, everyone. So maybe that might be your best next step following this. Take a look on there and see what you find. And I'm sure they would hope to see you in the sort of months and years to come. But other than that, hope to see you on our next webinar, and have a wonderful Christmas, everyone, and a lovely break when you get there. Thanks very much.
Thank you all, and hope you've enjoyed it. Hope you've learned something, and hope to see you in Leicester.